Today we have Min-Yao from University of Cambridge. Thank you for coming, for start your day so early to accommodate the time difference. And she's going to share with us the Xenium in situ and application in Medicago FFPE sample and how to use that to study nodule organogenesis.
Hello, everyone. Today, very excited to share our experience using Xenium in situ and how does this help us to understand the legume nodule development. And I'm going to start with the big picture about why we want to use this technology and how does it help us solve the question. And also going into a bit more detail on how is our optimization process. So if you are also interested in the technology, hopefully this will be helpful for you as well. So let's get started. So we are part of the NSAP project, which is the Enabling Nutrient Symbiosis in Agriculture. We are trying to engineer the crop species to hopefully generate the nitrogen-fixing cereal. And this means that in the natural environment, we have this association between the beneficial rhizobia with the legume species. So they can form this specialized structure called nodule that house a nitrogen-fixing bacteria. And this is a very specialized interaction between plant and microbes, so it's not existing in all different species. So we are trying to transfer this capability from the legume species to the cereal crops. So hopefully in the future, then we can generate this cereal crop that doesn't need to externally apply the nutrients fertilizer. So what is a nodule? Nodule is a specialized organ that can house this nitrogen-fixing bacteria and then provide an oxygen-regulated environment. So this is because the enzyme that's involved on the nitrogen fixation process is very sensitive to oxygen. So a tightly regulated environment enables the nitrogen fixation to occur. So why do we need to use the spatial transcriptome technology? So in terms of understanding the development of nodule, where and when the gene expressing and also their spatial content is very important to understand their developmental trajectory. And in terms of the engineering perspective, it's also equally important because as also mentioned in the previous talk, that if you use a constitutive overexpression or a knockout mutant, it's a good tool to help you understand the gene function. But in terms of the engineering project, oftentimes this important transcription factor also involved in either root development or shoot development. So if you use those methods, then you will have undesired side effects on other parts of the plant development. So here, understanding how is the gene expression pattern in terms of their developmental trajectory, specific time points, and also their expression patterns can help us to engineer the crop species in a much more precise way, introduce our target gene in the right time and in the right place, and also help us understand how do we rewire the gene network to enable this trait. And luckily, in the current data sets, we already have several single cell data set available in the Medicago nodule system. But the thing that's currently still missing is the spatial information. So during the process of single cell or single nucleus isolation, you isolated the individual nucleus or protoplast, which you lost the information of the spatial content. And this can make some specific cluster relatively challenging to annotate. And also at the same time, some specific cell type might be much more difficult to isolate from this preparation process. So then this comes to the spatial transcriptome technology. So in the introduction section, we already see quite a few of different platforms. And in generally, we can divide them into three major categories. And the first one is like the next generation sequencing based methods, so which is like Xenium that we already hear about. And then there's another two methods, it's in situ sequencing and in situ hybridization based approach. So these two are very similar. Similar also to the traditional in situ hybridization, the major difference is that now it's much more multiplex, and then using the fluorescent signal can help us decode the different transcript at the same time. So today I'm going to focus on Xenium in situ, which is an in situ sequencing-based method. And after the hybridization, they also have a rolling circle amplification to increase the signal to noise ratio, which make it a powerful tool to detect the transcript that have a low expression level. And that's also exactly the reason why we choose Xenium. And here is one of our bioarchive manuscript just recently made available. And then here cover of the process from the tissue to the data, from how do we prepare the sample, doing the sectioning, and then also the optimization for the probe design. So if you are interested in this optimization process, feel free to check out our bioarchive protocol. So here I'm going to give you a few of the highlights on the process that we are optimizing the protocol. So the first thing to consider is that different from the animal cells, plants have this rigid cell wall structure. And as you can see on the right-hand side over here, this is a Medicago nodule. And the plant cell wall, you can imagine it's also not a very homogeneous structure, different kind of cell wall type, you can have different texture. So those can also introduce some of the challenge. So here we are using a specific growth methods to growing the Medicago plant in the seed pouch. So then it's not growing in the soil directly. So it's not entangled with all the soil particles, make it much more easy for us to remove the plant material from their growing environment. And with this seed pouch approach, we can also observe the nodule formation in real time without digging them directly from the soil. And another thing to consider is the developmental stages of the sample you are working with. So for us, we are trying to capture the whole developmental trajectory from the very early developmental stages to the mature nodule structure. So we want to capture the tissue that at a very early stage before even the structure form. So having a method to be able to capture the sample before the structure form, we use a very precise spot inoculation method. So we can use a pole inoculation, put a 1 microliter droplet of the rhizobia inoculant, and then directly capture the specific root section for the following analysis. So in addition to considering your plant sample, another thing to consider is how do you prepare for the sectioning. So in terms of the Xenium methods, we can use a paraffin-embedded sections. And in this method, if you have a traditional in situ hybridization workflow that you know already work for your specific tissue type, it will be a wonderful starting point. Because for me, I have quite a lot of experience working on the traditional in situ hybridization, and this was very helpful for us to integrate this workflow directly into the Xenium workflow. Another thing to consider is your section thickness. So most of the animal tissue probably use 5 micrometer section thickness, but for a plant tissue, it depends on your cell size. So here you can see that the Medicago nodule not only have a different size of the cell in the plant tissue, you also see at the middle of this purplish color indicating the rhizobia is colonizing inside the plant cell and then creating this very loose and big cell structure. It also have a huge vacuole at the center, and all of this diversity can make sectioning a bit more challenging. One thing to consider when you are optimizing your section thickness is that the cell size. So if you have a much dense material like a meristematic tissue, the thinner section will be more suitable for your section. If you have a much loose cell type and then they have a bigger cell size, then a slightly thicker section will be better to maintain the overall structure. So once you optimize your section thickness according to different tissue characteristics, now the next thing to consider is your probe panel. So we start with small probe panel, which is a 50 gene panel to test at the mature stages how is a different cell type marker gene that's already been identified in a single cell data set, and how this is performed in the spatial transcriptome methods. And later we also expand the current custom panel up to 480 gene panel. We also included a design with a base panel and then an add-on panels. So it will be 380 plus 100s to make it much more flexible to swap to a different add-on panel if we have a different gene of interest in the future. We also designed the probe to make it suitable for multiple ecotype. So in Medicago, there's A17 and R108, these two different ecotype. It's commonly used. They are very similar to each other, but some of the gene, they still have a different sequence. And another thing to consider is the autofluorescence, which I will cover later. So the first thing that we consider is the mature stages with a 50 gene panel. We start by identifying the key regulator based on the time cross spoke RNA-seq, and then specifically identify the gene that are upregulated in the process after the inoculation, and then also the gene that are shown up in the central hub gene in the gene network. Those will be the gene that are more likely to involve in the key regulatory process, and also integrated the cell type marker gene already identified in the single cell data set. So this is one of the example for the Xenium. So we can see each individual dot here indicates a transcript, and then we can directly visualize them on the tissue contents. We can go from the whole organ level to the tissue level and to the subcellular level. And here we can see one of the example is NIND, which is a nodule inception gene, a key transcription factor involved in the nodule developmental process. We can compare with the traditional in situ hybridization. They have a high expression pattern in the, we call it infection zone, which is the rhizobia start to infect the plain tissue. And on the right-hand side, you can see the much more detailed structure in the subcellular level. This dark part is the vacuole. And then we can also see they have much more expression in the cytosol, the gray area, and also in the nucleus. The cytosol part also now colonized by the rhizobia. That's why it looks gray color. And compare between the infection zone and nitrogen fixation zone. So nitrogen fixation zone also have some expression, but much lower density. So now we are able to visualize multiple transcripts at the same time. It have an advantage to help us identify specific cell type and cell states, because oftentimes these are not determined by a single gene, but a collective of genes. So with this specific cell type marker gene, now we can identify different zones of the mature nodule structure. But something also caught our attention is that not all of the cell type marker gene that are previously identified in the single cell data sets are showing us exactly the same expression pattern in our spatial transcriptome data. And there is two potential explanations for this. The first one is a lot of previous cell type marker gene are being identified using promoter Gus assays. But the promoter can be an arbitrarily defined region. So if you have 1 kb, 2 kb, 3 kb, but if you have a cis regulatory element that's falling outside of your arbitrarily defined promoter region, then you might have a different expression pattern. Another thing to consider is the developmental stages can also change the expression pattern. So here is a clear comparison between these two. This is also a mature nodule, but a much younger nodule structure. So you see a round shape. And on the left-hand side, we are using a much more developed, elongated nodule structure, which is 28 days after the inoculation. So this highlight exactly the question we are trying to address. Only having the mature nodule structure is not sufficient. We want to cover the earlier developmental stages as well, hopefully covering the whole developmental trajectory. So from the very early infection stages to the mature stages. So one thing that I mentioned earlier about the probe optimization is one of the important elements is the autofluorescence. So if you are working on plant tissue, you probably know that the plant cell wall can have a lot of different autofluorescence depending on what type of cell wall you are working with. Another thing to consider, if you're also working with microbes, your microbe might also have autofluorescence. So in our case, both the bacteria and also the plant cell have a quite strong autofluorescence in this blue channel. So after our first round of xenium, then we see this very strong autofluorescence kind of block the blue channel, which can make the decoding a bit more challenging. So then we optimize the probe design in our later probe panel to try to reduce the usage of the blue channel, but still sufficient to do the decoding. So here we can compare between the 50 panel, which is not optimized at all, and then compare with the 480 gene panels. Now we have the high quality transcript detection rate increased from 75% to about 95%. So we are very happy with this optimization. And now we are working continue with the different developmental stages. Another thing to consider is that the spatial transcriptome result not only can be used as a qualitative analysis, it can also be used as a quantitative analysis. So here, after you have the image acquired and then you have the transcript information, we do the cell segmentation to define the cell boundary. And then we can quantify how many transcript is within each individual area. So then you can have a quantified result similar to a single cell data set. So on the left-hand side, you see the UMAP image, each individual dot indicating a single cell. But now this is the information based on just the spatial transcriptome data from Xenium. At the same time, you can see we can cluster the cell based on their gene expression profile. And now the advantage is that you can directly map them back to the two-dimensional tissue contents, much more easier to do the cell annotation and know which are the cell coming from. We are also currently moving forward to cover different developmental stages. This also gives us a benefit of doing a true time course spatial transcriptome analysis. So one thing to consider is that here we are highlighting different time point with a different color, shade of color. So on the left-hand side, we have the lateral root development. On the right-hand side, we have the nodule development. We start from the darker color to the light blue color, indicating from the 1 day, 2 day, 3 day to 4 day post induction, and similar for the nodule as well from 1 to 4. And at the same time, we can combine them together to compare how is their developmental trajectory changes and compare between lateral roots and nodule development. One thing to highlight is that this gives us a true time course analysis on their developmental trajectory. And then they also overcome one of the limitations in the traditional single cell analysis using the pseudotime analysis, which can be limited because they have the assumption that the developmental trajectory is linear or linear branching. But when we are looking at a real biological sample, it's not always true. Another thing to highlight here, you can see specific cluster that only have the red color or only have the blue color, which showing us that specific cell type showing up in only the nodule development or only in the lateral root development. So for us, the specific cell type that are showing up in the nodule development are those cell type that we most interested. So now we can focus on the specific cluster and then looking at the key marker gene and then look into what is their expression pattern as well. So today I just give you a short summary and a snapshot of how is the process we use the Xenium approach into our specific tissue type, and also the optimization process that we use in this first trial in the Medicago tissue. So at the end, I would like to thank a lot of people's help. So first, thanks my advisor, Giles, for his very generous support on this project. And also thanks for 10x Genomics and also the Cancer Research Center group to help optimize this protocol to suitable from animal tissue to the plant tissue. And at the same time, today I'm focusing on the nodule development, but engineering the whole project actually takes a lot of effort and engineering from a different perspective. So we also have collaborator that contributing to their gene of interest to our current panel that focus on different aspects of the nodule development, including the infection, signaling detection, and also the senescence process. Okay, so thank you for your attention, and I'm happy to take your questions.
Wow. Thank you. That was absolutely stunning. And like, thank you for presenting it also so clearly. It's just, yeah, the images were amazing. And
Yeah.
the work you went through to, the optimization and I think this bioarchive preprint, I think will be like cherished by many plant researchers. I have a question. So because with Jeff, when we are jumping on calls and talking to many researchers, now it's a lot of questions around detecting bacterial transcripts on Xenium. So I'm just wondering whether you included, just in case, any bacterial genes in your panel, and whether you detected anything. Just curiosity.
Not yet. So I'm also very curious about detecting it, the bacterial gene as well, because that's one of the reasons why we designed it to be a two compartment of
Yeah.
the panel design. So we can easily swap the 100 gene that's currently plain tissue targeted to the bacterial gene. So then hopefully we can see at the same time the plain transcript and the bacterial transcript. So I haven't tried it yet. We are currently trying to finish our current project with the different developmental stages and also some of the mutants. Hopefully the next one, then we can try the bacteria if it's possible.
I actually wanted to add that this panel design strategy, I just love it. Like, it's really so smart. LibG, well, we are waiting, just reminding the audience, feel free to ask more questions. We have a question, or actually it's a comment from Marta.
Marta.
From Marta.
So
Thank you,
when
Marta.
Jan was mentioning Marta, it wasn't this Marta. We have
Bali.
other Marta.
Yeah,
Marta Perez-Lobet,
Bali person.
who did Visium work. So Marta is saying, great work, Jan. It's looking fantastic.
Yeah.
I've got a quick question, if you don't mind, Marta. This Marta, not the Marta on the
This
line.
Marta, yeah, this Marta, yeah. I don't
Yeah,
mind.
you obviously used your single cell data set to help curate what genes you wanted to put in a panel, but have you also used your single cell data set to do label transfers to help better annotate the Xenium data as well?
Yes, this is a very good question. Let's come back to the observation. We see several of the cell type marker genes are not exactly the same when we see where they express. So now I'm doing two ways. So one way is that we analyzing the single cell data sets using what is the conventional cell type marker gene that label the single cell data sets. And if we do a label transfer to the spatial, can we see the matching labeling? But we also do on the other direction. So now we have the spatial information, also the tissue distribution, right? So it's easier for us to see where they physically located on the tissue. So then this gives us even more detailed cell type labeling. And if we do the other way around to do the label transfer in the single cell data set, can we see also the same cell type, or we can start to see a different kind of cell type? So this is a currently ongoing process. I guess another challenging, that's how is the integration process for the single cell and then the spatial transcription technology? That's also another thing that's haven't been explored as much. I mean, there's some label transferring methods by finding specific anchor gene, but can we directly integrate them together, become a single UMAP, and then can interpret them together? That's another challenging thing.
Yeah, that's interesting.
Yeah. Okay, 10X will have some work to do, I guess.
I also have a quick question to ask about the probe design. So since you designed the probe to target two ecotypes, what is your tips for us to be able to apply that to other studies?
Yes, so because these two ecotype, they have quite a lot of gene have shared gene sequence, so they have a conserved region. So usually those will be a good region to target. Then you can have the probe design they can target both of the ecotype. But you also have some of the genes that have sequence much more diverse between these two different ecotypes. Then you will design some of the ecotype specific probe. So in our case, that we have the gene, they are able to find 8 ideal probe for the conserved region, then those will be ideal set. And we also have some of the genes, they are much more challenging to find the shared probe. Then we will design an ecotype specific probe to make it up to 8.
Yeah. There's a lot of good tips here to apply Xenium to plant.
Yeah, so
thanks for sharing.
Thank you, for asking that question. Was just checking if there are any more questions from the audience. While I'm checking, it also worth mentioning, as if you're super inspired by our speakers today, we have some spring promotions running around. Spring is in Australia, so feel free to reach out, and we with Jeff, we are always happy to have discussions. I guess if we don't have any more questions, we can wrap it up. Thank you so much for presenting this absolutely beautiful research. And I will try to apply this knowledge in my garden.
Behind you.
Thank you.
Hello, everybody. My name's Jeff McDermott. I'm a Science and Technology Advisor with 10x Genomics, and I just want to start by saying thank you so much for being here with us today. We really do appreciate the time that you've taken out of your day. I just have some really quick slides to showcase the 10x Genomics product portfolio, which can, of course, be utilized within the plant and broader agricultural research. The slide just contains some important information about forward-looking statements that I may be making today. So to make any progress in unraveling the complexities of biology, you're going to need the right tools at your disposal. And you need technologies that provide insights at that fundamental resolution of biology, which is, of course, the resolution of a single cell. And it's that need that underpins the direction we take when building our products at 10x Genomics. And as you can see, we built 3 platforms that each allow you to take a look at single cell biology with a unique perspective. So we have the Chromium single cell, which, as the name suggests, enables high throughput single cell analysis, allowing you to profile transcriptome-like gene expression and other molecular features at single cell resolution for deeper insights into cellular diversity and function. Next, we have Xenium Spatial Transcriptomics, and this provides spatial whole transcriptome gene expression mapping at single cell resolution, allowing you to explore tissue architecture and study gene activity within the precise spatial context at tissue section level. And then finally, we have Xenium In Situ, which delivers in situ high-plex RNA analysis directly within the tissue sample itself, enabling subcellular localization and quantification of hundreds of targets for advanced cellular profiling and spatial biology studies. This slide, the numbers on here are a little out of date. They were put together in September of 2024, but what you see here are plant-focused research papers that have utilized at least one of the 10x Genomics products that I just introduced. So as you can see here, by this ever-growing body of work, all 3 of our 10x Genomics platforms can be used with plant material. Without diving too deep into the literature, I just want to showcase some common examples of why researchers have been utilizing the platforms from 10x Genomics in their plant studies. Shown in these next few slides are the main advantages of using single-cell and spatial transcriptomics in plant studies. I'll begin with studying cellular heterogeneity with precise resolution, and plants obviously have highly heterogeneous tissues where different cells within the same tissue or organ can behave differently depending on their location or developmental stage. So single-cell transcriptomics allows you to analyze the gene expression profile of individual cells, and by isolating and profiling individual cells, it becomes possible to identify rare or specialized cell types, such as those involved in defense mechanisms or nutrient uptake that would otherwise be masked in bulk RNA sequencing data. You discover rare and novel cell types, and single-cell transcriptomics has led to the discovery of new cell types and states within plants that were previously uncharacterized. These may include intermediate cell states during differentiation or rare cell types involved in specific physiological processes. And then spatial transcriptomics can be used to complement single cell findings by showing you where these novel cell types are located within any given one tissue. We're mapping gene expression in the tissue context. So speaking of spatial transcriptomics, this technique provides a 2D view of gene expression by maintaining the tissue architecture. It helps you as a scientist to understand how genes are expressed in specific spatial regions of plant organs, such as the leaf or root or stem, in relation to one another. This is important for studying how environmental signals like light and temperature and pathogens affect gene expression in different parts of the plant. Unraveling gene regulatory networks, and by analyzing gene expression at the single cell level, it becomes easier to construct these gene regulatory networks for specific cell types or regions within the tissue. These networks help you understand how different genes interact and control plant development and physiology, and this information is vital for identifying key regulators of important traits, such as disease, resistance, or growth rate and reproduction. We have understanding developmental processes, and plant development involves complex interactions between different cell types and layers within the plant tissue. So spatial transcriptomics allows for the mapping of gene expression patterns across the tissue whilst preserving their spatial arrangement. And it's really particularly useful for studying processes like root development, leaf differentiation, and floral organogenesis, where the position of a cell in relation to others is crucial for its function. Exploring plant environment and plant microbe interactions. Plants are highly responsive to environmental stimuli such as water availability, light intensity, and pathogens, as well as nutrient levels. So single cell and spatial transcriptomics allows you to understand how specific cell types or tissue regions respond to these external signals. For example, the response to drought stress in root cells can be profiled at much finer resolution, enabling identification of key regulatory genes and pathways involved in this stress tolerance. Gaining insights into plant reproduction, spatial and single cell transcriptomics are particularly useful for studying reproductive organs, flowers and fruits and seeds, and that's because these tissues often contain highly specialized cells. And understanding the gene expression patterns in individual cells during key events like pollen development and fertilization and seed formation can provide insights into plant breeding and crop improvement strategies. And finally, improving crop traits with high throughput functional genomics. And both single cell and spatial transcriptomics can be scaled up to perform high throughput analyses of gene expression across multiple plant species, tissues, and conditions. And this provides a comprehensive view of plant gene function and its regulation across diverse biological contexts. So these technologies can be applied to crop species, enabling you to pinpoint the genes responsible for desirable traits, such as increased yield or stress resistance or even nutrient content. So hopefully it's clear just from those few snippets of data that single cell and spatial transcriptomics is transforming plant research by providing high resolution insights into the complexity of plant biology. These technologies allow you to explore the diversity of cell types, gene regulatory mechanisms, and environmental responses at unprecedented resolution, opening up new possibilities for understanding plant development, stress responses, and improving crop traits. So if you've been thinking about applying 10x Genomics single cell or spatial tools for your plant research, or you're truly inspired by what you see here today with our speakers, then certainly don't hesitate to reach out. We'd love to have a project discussion with you and see what's possible. So with that, again, I'll say thank you very much.
That was terrific. See you next time in one of our webinars.
Thank you.
Thank you.
Thank you.
Bye.